We're going to turn our attention now to the richest source of information that we have these days about brains, which are the very many forms of imaging techniques that we have. And we're going to use the term image quite loosely to mean any means of recording changes, physiological changes or anatomical structures that can be then presented and interpreted spatially. So we're going to first of all distinguish between anatomical imaging and physiological imaging. Anatomy has to do with the structures that are in place. What can we identify as parts of the brain, for example? And physiology is a different enterprise. It has to do with what goes on there. It's usually interpreted in terms of functions, and functions will be dogging us at every turn, as they already are since our encounter with phrenology. Some of these means of generating images might not appear imagistic to you, so the analysis of what are called brainwaves, or EEG, is something we'll get to in a later video, but we'll group all such means of investigating brains together as imaging techniques. And we're going to start with an anatomical form of imaging, which is probably familiar to you. This is magnetic resonance imaging, and that's the scanner. Perhaps you've been inside a scanner. Many people have. They're absolutely routine in many clinical applications. They're a great way of obtaining detailed images about any part of the body. And indeed, they're not restricted to bodies. Scanners are used for investigating mummies, archaeological artifacts. They have the great ability to see through things, but they are not X-rays. Scanners don't use X-rays. The physics underlying the production of an image in a scanner is very complicated. Most people who interpret the images, who ask for the images, have no real grasp of how the images are constructed. The outer ring of the scanner contains large magnets which are made to spin very, very fast. They exert a very strong magnetic field, many times stronger than the ambient magnetic field that is everywhere on Earth. So the Earth's magnetic field is typically about 50 microtesla, whereas in an MRI scanner we might employ 3 Tesla, so 3 million times as great a magnetic field. This is why, of course, if you are going into a scanner, you must take out any metal, jewellery, implants and so on, because they'll be ripped right out of your body otherwise. It is, however, non-invasive. X-rays damage the tissue that they pass through, but MRI scanners don't do any damage. They generate images which are of great accuracy and spatial resolution, but they take a while to do that. It's a slow procedure, it's noisy. Subjects in the scanner are not allowed to move, but it does them no harm other than the slightly odd feeling of being stuffed into a tube. And the images that we get out tell us nothing about function. These are anatomical images, they resolve structures only. So here is one classic image that we might get in an MRI scan of a brain. Now the scanner actually records 3-dimensional images, and so we have to choose subsets in order to view them. Here we've taken what is called a transverse cross-section. Like as if you slice the top of the head off, you can see the eyeballs staring out googly-eyed from the front. But we could look at the same data, for example, with a sagittal cross-section, which is like this. You can clearly see the brain, you can see the nasal passages, you can see an awful lot of detail in there, so both brain and both bone, of course, but also soft tissue is very, very well imaged. And so we had a transverse section, we have a sagittal section, that leaves one, which is the coronal section, like this, straight down. And there you can see the ventricles in the middle of the brain. That's that T-shaped white patch in the middle of the brain. So MRI images tell us about the structure of brains, and they're very important. One thing to note about brains is that they differ greatly from individual to individual. At a coarse level, they have the same structures. So the hemispheres, the place of the cerebellum, the deep structures, those are identical across most neurotypical human individuals. But at a fine detail, if you get down to the level of a cubic millimeter, there's no correspondence from one brain to the next. Even at quite a macroscopic level, those characteristic folds that you see on the outside of the brain, they differ somewhat from one individual to another. So we're going to move from anatomy now to physiology using the same technology. This is functional magnetic resonance imaging, is a different kind of imaging technique. It uses the same basic principles as MRI, and it uses the same device as MRI. But what's recorded is not anatomical structure. What is recorded in functional magnetic resonance imaging is changes in regional blood flow through the brain. The signal that is picked up is generated by oxygen-rich blood, so hemoglobin, which binds with oxygen. And so the signal is called the blood oxygen level dependent signal. We don't get any absolute measure of regional blood flow in the brain. Rather, we get to observe changes in local blood flow in one experimental condition compared with another. And in order to orient ourselves and to know what part of the brain we're looking at, we first have to do a structural MRI. That means to understand the precise anatomy structures of the individual subject. Now, functional magnetic resonance imaging shares with MRI the fact that it's very good at resolving small spatial volumes, but its temporal resolution, its ability to spot rapid changes or slow changes, is absolutely lousy. It has a temporal resolution of about 5 seconds. So anything that happens in a period of 5 seconds is all going to look the same to this. Because it measures only differences in regional blood flow, functional magnetic resonance imaging is tied to the experimental situation. MRI is just a way of getting a picture of what's inside your head. Functional MRI is a way of imaging changes in blood flow as a result of experimental contrast. So in a very simple case, you might compare someone in a scanner with their eyes closed and the same person in a scanner with their eyes open. By employing the fMRI technology, you get to document the change in regional blood flow across these two conditions. So a typical procedure looks like this. First of all, we orient ourselves by doing a high-resolution anatomical MRI scan. That takes about 15 minutes. Then we conduct the experiment in which there are at least two conditions. Usually it's more complicated than that. But what we'll do is we'll do multiple scans, maybe about 150 fairly low resolution scans. Each one takes about 5 seconds to do. And so we can't make any distinctions within that 5 seconds. And we would do half of these scans in one experimental condition and half of these scans in the other experimental condition. This necessity for repetition severely limits what kind of questions we can ask, because we can only ask questions that can be answered in this way. There must be something that is stable across the experimental conditions, but obviously there's a lot that changes. Having done multiple scans in each condition, we can then compare the blood flow, that's what I've written here as activation, on a point-by-point basis. Now, in a digital picture, you're familiar with the pixel. That's the individual point out of which digital pictures are built up. Well, MRI and fMRI are dealing with three-dimensional volumes, so we use a unit called a voxel. That's a small cubic-like unit. Instead of a square like a pixel is, it's a cube. And we compare them voxel by voxel across the two conditions. So let's illustrate that. Here's first a complete MRI scan of a subject's brain, and then we conduct an experiment. And let's just say, for example's sake, in one experimental condition, you're speaking along with a recording of someone saying a sentence. And in the second experimental condition, you're speaking along with an actual person. This is just happens to be an experiment I was involved in. Just a small note, I did mention that you're not allowed to move in the scanner, but the slow process of capturing the changes in blood flow means that you can speak for a few seconds and scan for the few seconds afterwards while you're still, and in this way we can record the distinction between speaking conditions. It does require very, very many repetitions, and while we're scanning, you can't move. After that, we do the voxel by voxel comparisons, and here you can see, on the basis of this brain, you can see the colored ink that illustrates the differences in blood flow between the two experimental conditions. The way this is reported is shown down below. Regions that showed greater activity, that means blood flow, during synchrony with a live voice is compared with synchrony with a recorded voice. And we can see that there are differences between these two conditions. That turned out to be fascinatingly interesting, by the way. But there's a lot of important things to note about these images. They have a seductive appeal. It looks like you're looking at a brain, doesn't it? But if you don't know the very many steps that go into assembling this image, you could mistake this for some kind of a photograph of a brain, and it is nothing like a photograph. So these images have tended to be grossly misrepresented. The first thing to note is that they don't record any neural activity at all. That may seem surprising if we're calling them neuroimaging techniques. All these images can show are changes in regional blood flow. And then the language used to describe such images is entirely misleading. We often speak of the areas in the brain lighting up. No area in the brain lights up. And this whole notion of activity refers to changes in regional blood flow. So the interpretation of these experimental results is extremely difficult. So when you're reading any report of fMRI experiments, functional magnetic resonance imaging, you need to bear some things in mind. The averaging over time limits the kind of question you can ask. That means something has to be repeatable 75 times in a row. Now, your perplexity, your ingenuity, your creativity, your experience of the world is not of that nature. So we are limited in the kind of questions we can ask. Also, because brains differ greatly from one subject to the next, when we try to compare results across subjects, things get a little fuzzier and our ability to be precise in our spatial location goes way, way down. Blood flow itself is very difficult to interpret. We know it's related somehow to what the brain is doing, however that means, but the link between regional blood flow and neural activity is not yet understood. So that greatly limits our ability to say with confidence anything about what brains are doing based on fMRI. It doesn't stop people wildly exaggerating claims, but if you know the caveats, you'll be in a better position to understand what is and is not shown in such a scientific study. And finally, the last thing is to bear in mind the caution of phrenology. Phrenology assumed it knew what functions the brain served. It knew them to be distinct, and we laugh at it now because we say you had no right to assume that this innocent lump of meat was performing the function that you pulled out of the air. But we persist in doing this. The association of function with a place in the brain is one of the really problematic features of fMRI research. When done cautiously, of course, this is a sensible way to proceed. But when done incautiously, it's easy to slip down the slope back to phrenology. So we need to remember that anytime we attribute a function to a place in the brain, extreme caution is needed.